siempre la doctora Lara en aquí. Veamos. Ok. Okay. Vamos a ver. Listo.
Hola, ¿cómo están? Muy buenas tardes. Muchas gracias por acompañarnos una vez más a uno de estos jueves de educación continua, coordinado por el Colegio de Microbiólogos y la Comisión de Educación Continua. En esta tarde tenemos una charla muy interesante y se trata de la validación y requisitos para implementar el uso de reactivos recombinantes para la detección de endotoxinas bacterianas en sus laboratorios. Esta charla va a estar entregada a nosotros de parte de Timothy Francis, quien es máster en biología. Welcome, Timothy. Thank you so much for joining us today.
Thank you so much, and thank you all for joining this presentation as we talk today about the requirements for validation of a recombinant bacterial endotoxin test for your laboratories. This is a topic that has had much change, but also many things have not changed as fast as should be hoped. In terms of bacterial endotoxin testing and animal-free reagents. But what I would like to do today is provide you with a current update and give you examples with the recombinant reagent that my company has and examples specifically with that reagent in its use, and what it would look like to bring your laboratory to a recombinant bacterial endotoxin test as opposed to the traditional LAL reagent. First, I would like to provide an introduction to our product, Pyrostar Neo Plus, which is a recombinant cascade reagent that has all three factors of the horseshoe crab recombinant cascade have been produced. The terminology I'm going to use and has been used in some areas of this field is that of recombinant LAL or RLAL. This just refers to the reagents that are created that synthetically that are horseshoe crab free. Another advantage of these types of reagents is that they do not have factor G, and therefore they do not react to beta-1,3 glucans. So those are some advantages of all of these recombinant cascade reagents. From that, there are two varieties of these recombinant cascade reagents. The first is factor 1 reagents, referred to as RFC. These involves reagents that just have that protein that binds to endotoxin with those sushi regions with a fluorescence output. You can see the examples of the manufacturers of these reagents. We're going to contrast that with the 3-factor recombinant reagent, which is what I'm presenting today, in which what is called the factor 3, which binds to endotoxin, that then activates factor B and then a proclotting enzyme. A chromogenic substrate is added, which is then reported by absorbance readers. The current status of validation for these recombinant bacterial endotoxin reagents is not harmonized. I'm going to give examples from the European Pharmacopoeia and then the United States Pharmacopoeia. In the European Pharmacopoeia, the 1-factor recombinant reagents has been added to their chapter on bacterial endotoxin tests. I don't have this in my presentation, but the European Pharmacopoeia has expressed a new chapter that will be coming out soon, chapter 5.1.13. In this chapter will include the mention of the RCR, the recombinant cascade reagent. In the United States, which I'm going to focus on the USP chapter from this point in the presentation, both of these methods, the 1-factor and the 3-factor recombinant reagents, are in a new chapter, USP 86, alongside the traditional bacterial endotoxin test chapter, which is chapter 85. Just a general description of our product, as you can see here. It works by the kinetic chromogenic assay methodology. The methodology is virtually indistinguishable from the kinetic chromogenic assay found in chapter 85 of the USP over the bacterial endotoxin test in the harmonized pharmacopoeias. You can see the quantitative range, the quantity of the test. We've reformulated this reagent once to provide better suitability with certain products, especially heparin, as well as a better distinction between the negative control and the low point of that standard curve. The other 3-factor recombinant reagents are very similar in form and function to this reagent as well. And these recombinant cascade reagents all work by the kinetic chromogenic technique, just as kinetic chromogenic LAL reagent. What I want to do is look deeply into what this chapter, USP 86, which in terms of the timeline of this chapter, it was released in May of 2025. You can see a history of the US FDA and the US USP timeline in proposals, different advisory chapters, and then finally the acceptance of this chapter, which has a mention of these items. One other current update I'll provide to this slide, although it's not official, the United States Pharmacopoeia has released 13 monographs for public comment on the different waters that require the bacterial endotoxin test. For example, water for injection, water for irrigation, water for the processing of medical devices, water for dialysis, have monographs in the process and in review that will be added, and basically then this method would be fully compendial and then referenced in the monograph for those methods. However, I'd like to discuss what the current situation is in using a recombinant bacterial endotoxin test method, such as Pyrostar Neo Plus, for all of your bacterial endotoxin test needs, from in-process testing, screening of raw materials, all the way to the FDA submissions or the submissions to the appropriate regulatory body. The terminology that has been used to describe this is a compendial alternative. Now, in some ways, that's an oxymoron, as compendial and alternative mean almost the opposite thing. But in reality, what this is saying is it is compendial, as it is now listed in the USP compendial chapters. So the USP has already reviewed the primary validation, and accepted these methods as bacterial endotoxin tests. However, they have not yet published specific monographs for specific products. So for a product that currently has a reference in its monograph to, for example, USP 85, then the user would have to provide the demonstration of USP 86 is alternative to that. This has had a great success in the United States, and I think of certain companies such as Eli Lilly. They've recently reported that 70% of their end products have been converted to some form of recombinant bacterial endotoxin testing. So this is something that definitely the FDA and the USP are working for. But what a submission would look like would be very similar to the suitability testing that already is needed for the bacterial endotoxin test, with three key additions potentially needed. The first is the manufacturer's primary validation package, which is provided. This is the validation that the USP has already reviewed and we would provide for you. Second is a potential for an orthogonal method study, which in this case would easily be performed by comparing traditional chromogenic LAL reagent on the same equipment with this new Pyrostar Neo Plus, this three-factor recombinant reagent, which I believe is a great advantage of this reagent, in that it is really easy to reduce the number of variables in performing this test, as it uses the same kinetic chromogenic method. This reagent also can be reconstituted with water for injection. It doesn't need a beta-glucan buffer, and so therefore this reagent can be very easily in a laboratory setting be used to give side-by-side results with the traditional bacterial endotoxin test method. And then the chapter also mentions that regulatory bodies may require additional information as needed. I will show you some questions and answers in a moment that essentially outline for us that this most likely will be examples of the reporting of the bacterial endotoxin from your naturally occurring endotoxins at your site, for example, from your DI water loops, just that the endotoxins that you are typically experiencing are being reported by this reagent, just as it is by the traditional bacterial endotoxin test. So with these additional pieces of information, the compendial chapter, chapter 86, says that this method then is suitable for being substituted with USP chapter 85. So this just provides an example of what this looks like. Essentially, we no longer have to perform an alternative method validation that already has been performed and is referenced with the primary validation package from the manufacturer. And then the suitability testing for the product, which of course is the preparatory tests found in virtually the same as they are found in USP 85. Plus any additional information as we just mentioned. So you can see the conclusion there is that this test can be substituted out for that compendial method. What I have here is a document that was actually released by the USP, so this isn't my work. You can see the link at the bottom of this page. It's freely available for anybody to look up, but this is a very useful document which gives some clarifications on what the United States Pharmacopoeia is thinking with these methods. So some of these are just definitions, as we see here, a statement of purpose. I've given much of this information here. Basically confirmation that the USP has evaluated these reagents, these recombinant cascade reagents that was mentioned. Question number 6, as you see here, provides that information that I'd mentioned about the additional information that may be required for the customer or the client, I should say, to provide that testing information. And then one other item as well, and I believe it's in question number 5, is that the same reference standard endotoxin that is used to validate the traditional bacterial endotoxin test is also applicable for USP86, these recombinant reagents. And so, of course, that is something that simplifies it for us as clients, as the pharmacopoeial bodies provide this reference standard endotoxin that they now say is suitable for us. This simplifies things as opposed to, for example, using a monocyte activation test where standard non-endotoxin pyrogens are not as easily harmonized and acquired. This document also provides just an overview of what the different pharmacopoeial bodies and their perspectives are. This document was released at the end of 2023, so some of this is out of date from the answers or the updates that I gave at the beginning of the presentation. And then I apologize, I had misled the question I was referencing was question number 11, where we do get confirmation that the USP's reference standards do apply to the bacterial endotoxin test. Thank you for allowing me to go through those questions very quickly. As I mentioned, this document is freely available, and if you didn't have time to copy down the link, just a Google search of USP Chapter 86 questions and answers, I believe should bring this information up for you. All right, so this concludes the first part of my presentation, and thank you for bearing with me through this. We went over much details in terms of what the regulatory situation looks like, and I wanted to go through that because, of course, I think for many of us, we want to move to a new method, especially a method such as this, where there is much sustainability goals that this reagent applies for. But there are questions, of course, with the regulations. And if you would like information about that, I'm happy to provide examples of organizations in the United States and then also in other countries as well, where the United States FDA has accepted the USP 86 chapters for the bacterial endotoxin testing of their products. But what I want to do now is to show you what the operation of the Pyrostar Neo Plus would look like. I understand that I may have a varied audience. Perhaps some in the audience are currently performing the gel clot technique, and that's the only technique that has been used. Others may currently be performing either an endpoint chromogenic method or a kinetic chromogenic method, in which the practical application would look very similar. But to bring all the audience on the same page, I'd like to just give a basics of the form and the function, and then the application of this recombinant cascade reagent. Very simply, what we see here is exactly what I have mentioned about the 3 factors of these recombinant cascade reagents being synthesized. What we see here is actually a mapping of all of the bacterial endotoxin test methods. The 3 methods listed as gel clot, turbidimetric, and then chromogenic methods. All of these come down to endotoxin binding to factor C, we call it, which then initiates a cascade reaction. Factor C then cleaves factor B, which then activates the proclotting enzyme to become clotting enzyme. In the natural product, this forms a gel, which can be measured by the gel clot. That can be also inserted into an absorbance reader and measured quantitatively that way. And then also the PNA chromophore could also be cleaved. So that method, the chromogenic method, allows for a more enhanced reaction and therefore a more sensitive reagent for the bacterial endotoxin test. And of course, the recombinant cascade reagent, as we have discussed so far in this presentation, the Pyrostar Neo Plus, is essentially factor C, factor B, and proclotting enzyme. So those three factors have been synthesized. The chromophore has been added, the peptidyl PNA, and then of course the reagent has been formulated, buffered, and stabilized as needed. The method by which this reagent is measured is a kinetic method. For those of you that are only familiar with the gel clot technique, or perhaps an endpoint chromogenic technique, you are used to an endpoint method, essentially where after a predetermined amount of time, a measurement is observed, whether it is the physical measuring of a positive or a negative gel clot result, or of course, readouts from the reader allow for a response of standard endotoxin versus the intensity of that signal. Chromogenic methods work on a slightly different manner, and I hope this visual provides a demonstration of that. On the x-axis, we have measurement of time in seconds, and then on the y-axis, we have, from an absorbance reader, measurements of optical density. And so essentially this assay is a kinetic assay in which repeated measurements are made over time. As these measurements are made, of course, that endotoxin binds to factor C, but this is, of course, a cascade reaction. So there is some time delay, but eventually, of course, that intensity of the reaction takes place. We see an increase in absorbance, and we see that activation taking place. And the key item for these kinetic assays is that the actual reaction is the time of reaction is dependent on the concentration of the product. So you can see, for example, here, 4 standards that could be used to create a standard curve. And then what we would use is some threshold value to give us the activation time. You see here the threshold optical density is listed, and you have a confirmation that the negative control, of course, did not react. An advantage of a method like this is that the endpoint methods are often limited to a 2 log at most range of the standard curve, but really standards can continue to be added, and the time of measurement will allow for the distinction of that. So this just provides, the definitions that we saw of an endpoint method. Gel clot is an example of an endpoint method, basically a predetermined amount of time. It elapses at which point a measurement occurs. This is limited in the amount of information that can be measured, whether a simple positive or negative with gel clot, or a 2 log standard curve with the endpoint quantitative methods. But then the kinetic methods allow for a much broader range. And so this is all the all the quantitative reagents that my company provides are kinetic, and of applicability here, the recombinant reagent is a kinetic method as well. This is an example of that data that would come out based off of that threshold optical density. Once that threshold has been crossed, an activation time can be recorded. So now we have a data set of 2 variables. The concentration for our standard curve becomes the dependent variable. The activation time becomes our independent variable. We have a log-to-log relationship between these two, and so you can see a linearity can be produced from that. For those of you who are maybe not familiar with this form of data regression, there's no need to worry, as this is the standard data regression used by the kinetic endotoxin testing methods. And so as a result, there's software available and also protocols available for the various software that can perform that data regression for you. This is just visually showing the products that I work with that are that follow this principle. Our turbidimetric method, which also works as a gel clot reagent, our chromogenic reagent, and then the emphasis here, of course, is on our recombinant cascade reagent, that Pyrostar Neo Plus product. So what I have here are just simple pictures showing what this would look like. I understand the assay can have infinite number of iterations and what it would look like for you, but essentially every bacterial endotoxin test is going to come down to just 4 different types of samples or 4 different types of solutions that are used. There's the negative controls. The pharmacopoeial chapters refer to this as solution D. There's the standard curve or solution C. Of course, for the gel clot technique, that is my 2 lambda spiked standard. And then, of course, we have the sample and the spiked sample. One difference between the gel clot technique and the kinetic techniques is that instead of spiking the sample to a 2 lambda concentration, the sample is spiked in order to form the positive product control. It is spiked to the midpoint of the standard curve, and then recovery is based off of that. That's all normal and expressed already for us in USP Chapter 85. So this is no different from what has already been accepted with the bacterial endotoxin test and the recombinant reagent. So with that understanding, I could plate in triplicate, for example, the negative controls, a 3-point standard curve. I know I showed you a 5-point standard curve previously, but the standard curve only needs 3 points for a valid standard curve according to the guidelines, and then a sample, and then a corresponding spiked sample. Of course, I could have tested these in duplicate. This would have been perfectly valid, and I could have filled up this plate with additional samples and spike samples, additional samples and positive product controls. What that visually looks like, and I hope this comes through on the screen. It's a picture of a faint amount of color, but there is some color there. And especially for those in my audience who currently use the kinetic chromogenic reagents of the traditional LAL reagents, I just want you to compare that in the response of those reagents to what we see here. Of course, not much useful information can be given from this, but we see the negative controls here in these 3. We see these are colorless, so no reaction took place. We see that color that was produced here in the 3-point standard curve. I believe we had a 0.1, a 1, and a 10 EU per ml standard. And over time, of course, that color is produced, and then the sample, and then the PPC here. At least from my perspective, regularly working with the kinetic chromogenic reagents, and then now this Pyrostar Neo Plus, it's almost a comfort to me to see this, because it's exactly the same response as we're used to with the traditional kinetic chromogenic reagents. Of course, us visually after the measurement, we don't get much information from that other than wells that seem to be free of endotoxin and wells that have endotoxin in some form. And that's why I want to now go to what what we call the time course from the software would look like. Of course, a kinetic measurement takes multiple measurements. You can see those here. I know I don't have a legend on the screen for sake of clarity, so you can see the visuals much better. But here we have the samples as well as the negative controls unreacted. We have the 3-point standard curve in triplicate here, 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 and here. And then the spike samples, spike to the midpoint of the standard curve, would have been here. My threshold value, of course, is down here. But for those of you that visually have worked with either kinetic turbidimetric reagents or kinetic chromogenic reagents, we see something very similar to what you're used to for that on exactly the same reader. This is measured at 405 nanometers with a 37-degree incubation time, and it can be run on any microplate reader that can perform kinetic measurements with those parameters. I just wanted to contrast that with what a typical kinetic turbidimetric assay would look like. We see the same shapes, just as you'll notice, and this is the same for chromogenic reagents. Turbidimetric reagents often have a change in optical density of around 0.1 optical density units, whereas the chromogenic reagents give us a much nicer 0.5 to 0.6 jump in that. And so you can see the scale is actually much greater here. We see a much stronger signal with that. So that just provides a contrast of what a natural LAL reaction looks like versus here, the Pyrostar Neo Plus reaction. The data analysis, and this comes from our, we call it the FQC1 software. That is the DQ has had full compliance with the USP and FDA guidelines, and this is the software we use with the FDA licensing of our traditional LAL reagents. So these reports are FDA approved and are full compliance for your needs. But here we have the standard curve with those 3 concentrations, the activation times. This is a snip, if you will, directly from our test reports that we have. So no need to have these parameters set up customized in your software. This would work with the software that we have, as well as any of the software of the multiple other kinetic chromogenic manufacturers. This shows for us what the results would look like with these, the sample A, of course, no activation. So we're seeing a measurement of that. And then I asked the software to round for that. I didn't show you what the endotoxin limit would have been, but of course the software gives a judgment of pass for that. And then the positive product control, the measure of endotoxin for that, and then recovery rate, the criteria for that is 50 to 200%, and we see the measure of pass for this. So what I want you to picture, of course, is what would it look like to provide the validation for adopting a new product for these test methods. You would perform the test for interference, which is exactly what we would have seen here. Of course, the test for confirmation or assurance criteria of the standard curve, just as you would with the normal bacterial endotoxin test. A side-by-side study isn't necessarily required, but is key. That could be performed on the same plate. You could have, the bacterial endotoxin test reagent, the natural LAL reagent on one side. You could have this recombinant cascade reagent on the other side of the plate. Very nice side-by-side reports with that. And then also for a couple samples, you could have the different natural forms of endotoxin that you have acquired from your lab, if your regulatory bodies require that. Some may not even need that. This, in addition to that primary validation package that we will provide, showing all criteria that traditional LAL reagents pass in order for the FDA submission and licensing of the product. We show those same tests applied against our recombinant cascade reagent, and they pass as well for that. I mentioned the software that is used. Of course, one hurdle that many have is just the setting of the parameters, the data integrity for bringing in a test like this. Perhaps there are users that are currently outsourcing the bacterial endotoxin test to a testing laboratory, would like to bring that inside. And so that is why I would recommend a software. An example is the FQC1 software that I'm familiar with, but a software like this would give you something that is already used in FDA submissions and essentially sets the criteria for you and sets those parameters, that has full data integrity. And so you can see just the items that software eliminates as far as variables for you in terms of wanting to bring in this reagent. The software allows you to set different what are called test groups. So essentially making it possible to run on a plate, for example, 2 different types of reagents, because it's going to treat those as 2 separate assays, and it will give you 2 separate test groups based off of those well assignments. 2 different test reports will be created. That's the end overall of the presentation that I have. So my concluding statements are this. Much interest has been had in these recombinant reagents, and it has been a slow process for the regulatory bodies to bring this. But now here in 2026, we've reached a point where, although it's not yet listed in monographs, which once it is, that will be wonderful, because these reagents can basically be interchangeable with the bacterial endotoxin test reagents. But even now, for an end product, if a new end product is coming to the market and you would like to try this reagent, if you currently, for example, have an absorbance microplate reader, and you have protocols for the kinetic chromogenic reagent, essentially you would follow the same procedures you currently do for the test for interference, the preparatory tests for the suitability of that, in addition to a validation package from the manufacturer, which of course we already would have done and supplied for you, in addition to an orthogonal study, which is easy to do, especially alongside a kinetic or a chromogenic reagent, and then also potentially measures of the endotoxin in your sites. But with those, it becomes much easier to see a path forward with these types of reagents. So my concluding thoughts for you are, I would recommend if this is of interest, a phased approach. We already know the water monographs are coming to scale. So a risk-based assessment, become familiar with these reagents in your in-process tests, evaluating raw materials, and then once proficiency in ease of use is achieved with your team, which should be simple, as this is very similar to the traditional chromogenic reagents. But even for if you are currently a gel clot operator and you would like this type of reagent, then the software and the protocols that can be provided that are already used in FDA submissions will help guide you through that. And then, of course, the technical support from the Fujifilm team, definitely for this reagent, is provided along the way, as well as all other reagents. The technical support is there. So I want to thank you so much for your time and attention in listening to this presentation. I believe there's a time now for questions, so I will be happy to answer any questions that you have at this point in time.
Bueno, muchísimas gracias. Thank you, Doctor, very much for this interesting. We want to know if they have any questions, so I'm going to ask them. Si alguien del auditorio tiene alguna pregunta para el doctor, este es el momento. Bueno, mientras tanto, yo le voy a hacer una pregunta, doctor. Doctor, why would you recommend switching to recombinant reagents, and what are the advantages over LAL reagent?
Yeah, great question. Of course, the recommendation would be not necessarily something now to replace the traditional LAL reagents. That's actually one of the items in that USP frequently asked questions that I'd mentioned as it's not going to be replaced. The USP 85 test is still going to be the gold standard. However, the advantages of this would be the really for the assurance of your company in redundancy, in potential supply issues with, of course, naturally sourced LAL reagent. It would be good even if it's not something you move forward right now. It would be good to at least have a mastery of that if needed in terms of these synthetic reagents, just as a secondary source. And then as well, I do believe the price will come down as manufacturing is able to scale up for these reagents, and as they're able to be built on scales there. So I believe eventually, even though that might not be realized today, there will be cost advantages to these reagents as well.
Perfect, thank you. I also, what are the advantages of the 3-component reagent over the single-component reagent?
Yeah. The one factor recombinant reagents are reagents that have a fluorescence output. They're an endpoint technique, and this is a technique that's novel, that's new to the bacterial endotoxin tests. So even the endpoint methods from the traditional USP Chapter 85 are still absorbance measurements. They're still measured at the same absorbance wavelength. So one advantage definitely is that the equipment, the water that's used to reconstitute, the plates, the optical plates or tubes that are used, all can remain the same. And then another advantage from that, of course, we know from science, a principle is only change one variable at a time. And so if we need to go to a new method with all new equipment, new IQ/OQs need to be performed, new training, there's a lot of variables that need to be qualified versus I think one of the best advantages of these recombinant cascade reagents is they can actually be run on the same plate side by side with the same accessories, same water, and get equivalent results very simply in a practical setting.
Yeah, and since you are mentioning this, I was wondering, because you mentioned that no buffer needs to be added to the reagent. So does that mean that it's ready to use and you just have to add the sample, right?
Yeah, it's it does come in a lyophilized form, so a powdered form, but it is reconstituted with water. So no special buffers needed. That the same water that you're going to use, the same endotoxin-free water you're going to use to make sample dilutions, can be used to reconstitute the reagent. And with that, then the reagent will have buffering capabilities, and then with no factor G, there's no need to add an additional glucan blocker to make those results endotoxin specific. So it really is a simple procedure, just adding the water needed, swirling it for about 20 seconds, and then it's good to go.
Perfect. Ok. No sé si alguien del auditorio quiere hacer alguna pregunta.
Doctora, en este momento no tenemos ninguna consulta.
Okay. Well, then we have no more questions, doctor. So thank you very much for joining us tonight and for giving us this lecture. Very interesting. And so I guess this is all for tonight. So thank you very much for joining us.
Thank you so much for this opportunity. It's been a wonderful time presenting this to you all, and I hope you all have a good night. Thank you.
Thank you. Buenas noches a todos y muchas gracias por acompañarnos.